Hi, my name is Ethan Hill. I am the bioinformatician for the Iolani School Office of Community Science.
And I'm Jamie Frith, a senior taking honors independent research at Iolani School.
Today, we'll be presenting genome science in high school classrooms, inflammation, glutamate, and methylation. Gene body methylation has been shown to be a proxy for gene expression. It facilitates transcription, elongation, and modulates alternative splicing. It has also been used for personalized medicine in cancers, but has yet to be explored in inflammatory diseases.
Cytokine production is caused by inflammatory gene expression, and cytokines are signaling proteins that help regulate inflammation in one's body.
Glutamate consumption has also prompted inflammatory reactions in individuals with psoriatic arthritis, which is a chronic and inflammatory arthritis that occurs in relation to psoriasis. That leads us to today's topic, a case study of the relationship between glutamate consumption, psoriatic arthritis, and gene body methylation. Specifically, which inflammatory genes show differential methylation upon consumption of glutamate in a patient with psoriatic arthritis despite taking TNF-alpha inhibitors. So the general workflow for the sampling happened as such. Two patients, one with psoriatic arthritis and glutamate sensitivity, and one without, had their blood drawn 3 times, once at time 0, then 5 days following glutamate consumption, and a final collection another 7 days after that. PBMCs were then isolated from blood, DNA was extracted, and followed by MinION sequencing. A custom bed file containing 226 inflammatory genes was created for adaptive sampling. Methylated base aggregation was conducted using the WF human variation workflow through Epitome Labs. Differentially methylated genes were then identified using Wilcoxon sign rank tests. A similar method was also used for the cytokine analysis, which was conducted using a 17-plex human procartoplex panel. Cytokine concentrations were then also compared using a Wilcoxon sign rank test.
Regarding our cytokine results, we found a statistically significant relative change in the cytokine concentrations of both individuals after glutamate consumption. The graph on the left illustrates the 17 cytokines recorded from the panel, while the figure on the right represents the statistically significant relative change in cytokine concentration.
Of the 226 inflammatory genes screened, 150 showed differential methylation when compared to the baseline. 50 of these were unique to the arthritic patient, 23 were unique to the normal individual, while 77 were shared between the two.
And specifically for the arthritic patient, differential gene body methylation of some inflammation-related genes was observed following glutamate consumption. We saw an increase in gene body methylation of CCL4 and CSF3, while we saw a decrease in methylation for PTGER1 and MARS1.
We additionally saw differential gene body methylation occur in opposite directions for the following genes: CCL5, CCR4, ALOX12, and PTGS2.
Regarding IL-6 and IL-8, the figure on the left illustrates their individual cytokine concentrations at all 3 blood draws for both individuals. And here we can see after glutamate consumption, there is an increase in cytokine concentration for the arthritic individual, while we did not see this increase in the normal individual. And the figure on the right represents the statistically significant percent change in methylated CpG sites for IL-6 and IL-8 that we saw in the arthritic individual using a Wilcoxon sign rank test. However, we did not see this significant change in the normal individual. But more importantly, what we would like to point out is that the increase in cytokine concentration of IL-6 and IL-8 corresponded with the increase in gene body methylation, indicating an upregulation of both.
So, what we have seen here is that despite taking TNF-alpha inhibitors, the inflammation the patient experiences could be linked to the upregulation of IL-6 and IL-8. This means that IL-6 and IL-8 inhibitors could be used as supplements to the existing TNF-alpha inhibitors to control the inflammation. Additionally, the increase in gene body methylation in CCL4, CCL5, and CSF3 could also indicate other genes that could be contributing factors to the inflammation and may potentially aid in decreasing inflammation when suppressed.
We would like to thank those a part of the Mauna Kea lab at the University of Hawaii at Manoa for assisting our lab work, as well as those a part of Iolani School's research team for assisting our lab work and supporting us through this entire research process. We would also like to thank Hawaii Dental Services, the National Cancer Institute, and the National Institute on Minority Health and Health Disparities for funding this research.
Thank you.